and bolts of it is I took a couple of cell lines and reduced and alkylated a cell pellet, got a cell pellet, lysed them, reduced and alkylated, did the trypsin digestion, all the things that you guys do, week in and week out. Resuspended those in what we call our buffer A. And then I made up some 100 nanogram and 1 microgram aliquots to be able to inject. That's what I wanted to inject. And the way we do things here is we measure at the protein level, and then that's the concentration. We don't remeasure again at the peptide level. And then I measured these. So one of the first question I wanted to know is, because Aaron Story had started DIA here, and he had these staggered windows, and some people do non-staggered windows or don't do that. And so I wanted to know, is there a difference? And so just to kind of go in, so if you're in your thermo instruments and you pull up those red boxes here kind of show, you really got, there's a lot of information here, but this red box here just sort of shows this is where experiment number 1 comes into play. And that's the first experiment. It's going to be the MS. We're just going to do a scan of the precursors. And then experiment number 2 comes in here where you have all the DIA settings. And then these are the windows that we have here. And then here's some of the information as far as those settings for this. And the big ones is, what's your precursor mass going to be for your MS, for your DIA experiment, what's your window isolation, what your scan range is here. Mike talked about, I think you did like 380 to 9 something.
350 to 950.
Yeah, so his is a little shorter, ours is a little wider. We're probably not getting a lot of course anyway, these are the things that you can tweak.
That's my precursors.
Oh, okay, your precursors, yeah, up here, but I think we're still a little bit higher. Anyway, and then this guy down here, which I think you had to set to automatic, we've got it set to 40 milliseconds here. If you do a staggered experiment, the way we do it here is we've got a single MS run followed by the first DIA experiment, followed by another MS run, followed by another DIA experiment, which after we got into this, I thought, well, what if I were to take out that second MS run so that I had more DIA time, what is that going to tweak my numbers a little bit there? But you'll notice the stagger means that, of course, you can't see these numbers because they're so small, but we're starting at 394 is where a window starts for the first experiment. And in the second experiment, the window starts at 400. So it's just offset by about 6 Thomsons there. But other than that, the experiment's pretty much the same. So what I did to evaluate this is I put the raw data through DIANN, and the files were searched against the spectral library that was made from the human FASTA file. So that step was done first, and then I had that spectral library, and then I got this file. One of the output files is this PG, stands for protein group matrix, and it looks like this, and it's got your annotation over here, and then it's got your abundance over here. And I was kind of curious, it sounded like Katia had said that there was another table or something that had different values out of DIANN, but I'll follow that up later, other than these. And so using this product that Stephanie's going to talk tomorrow morning about, ProteoDA, which was developed here by the team of bioinformaticians that she works with. We come up and I could do this. Now I'm not bragging, but if you were to take 2 different cell lines and then two different methods, this is how you stack the cards against you. Could you get better groupings here than you have? Now, I was kind of worried to do this. I was thought, because it was like just a day or so ago, and I thought if this doesn't turn out and these things look all gnarled, then I'm not going to show any of this data. But this is part of our QC report that we give that we do on every project, and it shows basically your load over here in this what's that called, the violin plot, and then you've got your PCA, and then you've got your little dendritic map there, as well as these other things. So that all these plots are just provided so that you can look at, just kind of get a bird's eye view, which I think is really important to see how good is the data, how good was the experiment as far as hitting the goals of having equal loading, and you can tell a lot by the usually this PCA plot will tell you a lot from the beginning here. So the goal of this experiment was, how different are the significant protein IDs in a DIA experiment with staggered windows versus non-staggered windows? Okay, so I want to I do the experiment, now I want to figure out which proteins are significant in the relationship between HeLa and our JJN3, and then I want to have them in two different groups, the staggered and the non-staggered there. And so I'm just going to show you. So I ran this experiment on an Explorers 480. I injected 100 nanograms in every sample. And then I used, like I said, DIANN to do the searching of it. And I've got this picture up here both ways, because this really isn't very satisfying, because I don't know, Venn or these Venn diagram things that you can get off of the internet. I did this one as well because it shows you the relative amount. So it turns out about 61% of the significant proteins were identified either by the non-staggered or the staggered method. And when I showed that to Rick, I thought, is that right? Because that just seemed low. I thought, really? Rick was like, yeah, that's what we get.
My comment was, if he'd have run this experiment twice using either staggered or not staggered, just repeated the experiment, I would have expected very similar results to this.
How are the staggered spectra deconvolved?
I don't know that they were.
Oh, okay.
So how do you mean? Explain that term.
Yeah, so I mean, the whole idea of the staggered workflow is that you have overlapping regions in your buckets that can be virtually deconvolved, which normally happens in MS Convert. They have a demultiplexing option, but if that doesn't happen, then effectively it's not really a staggered workflow; it's an overlapping workflow.
Oh.
But I mean, I'm okay. But I'm glad you're looking into this, because I mean, the whole reason of having a staggered workflow is because the instruments at the time weren't fast enough to be able to do two Thomson isolation windows at whatever speed the newest instrument has. Sorry for interrupting.
No, that's good. So yeah, the nuances between staggered or overlapped, I guess we can get into that some other time. But I was trying to figure out in this experiment if, because the idea that Rick was talking about earlier about how on the edges of those windows or those you can have distortion, was that would we get more IDs if we used staggered? Would we get better coverage of that? That was my thought. And so we ended up with about 1,600 significant proteins using the staggered, and about 1,530. So those aren't total. That's just the significant ones that we found there. So these are the volcano plots for each one of those. The one that is the no staggered is over there on the left, and the one for the staggered is on the right. And even though that other number was like 61%, those volcano plots look eerily familiar, like or similar. And you can even see like some of the groupings up there. So those are that gave me a little bit more courage that our DIA data is that despite some of the you know, you want to be able to run the same experiment 5 times and get the same answer all 5 times, but there's so much data coming out of here. There's so much complexity that that's not as easy. It's not as simple as that. But that gave me some courage that it's actually working like it's supposed to.
Dennis, I'm gonna ask a question that I should probably know the answer to, but I don't. In this case, for JJN3 and HeLa, there are gonna be proteins that are detected in one and absent in another because they may both be present, but their dynamic range is far enough apart that they're gonna appear to be absent. How is that data handled in this experiment? Are those listed in the significant list or are they not? And I'm looking for Stephanie now.
So it depends on if you're taking the full output from the database search or not. So the overlapping, you have that unique and common set. So I'm assuming this is everything from both of those data sets. So if you have some identified in HeLa, then it's going to be a 0 in the other cell line. And so depending on how you run your lemma, if you're omitting the zeros, then your full change is different than if you leave them a 0. So it kind of depends on how we run our stats.
That's what I meant.
Yeah.
I didn't know how this was run.
I don't either.
Okay.
How would we know that?
I'm assuming you probably ran it with NA, so NAs are probably omitted from this, but if you
Okay.
click on the dots and the other plot is where it shows me that information. We can go through that tomorrow morning.
Okay. Yeah.
So what I did is I changed for this, when the output from DIANN, I can't remember if the output from DIANN just leaves blanks or if it has NANs. But anyway, I changed
If
all
you
those.
look at
zeros.
that report too at the bottom, it gives you the total protein IDs. If it's different, then you'll also know if it's
I see. Okay.
staggered versus non-staggered.
So this particular experiment, I think that, and maybe because we ended up with about 6,100 proteins identified for each, whether it's staggered or unstaggered. It turned out to be the exact same number of proteins, which that was something. Again, the goal is not to, the goal is for me to in doing this is to try to understand. And obviously I showed that I don't, because I use the word staggered when I should have said overlapped. But I will say a take home is it's a lot easier to run these sort of things when someone else has done all the legwork and just keep doing what they did, hoping with the understanding that they did their homework than for me to try to come up with this by myself. So that's the first vignette here. I don't know if I want to go through this one. This one's a little scary. Are you all right, Mike? Okay. Okay. This did not turn out as well as I would like. So we took this one's a little different. Instead of having the instruments all sitting down in the same room, I took some samples and sent them to Mike, and he ran them through his workflow. And then I did the same thing here. He used his QE that he's been talking about. And I think those are his parameters. There's 20 Thomson windows, and he's got 30 windows going from 350 to 950. I used an Exploris downstairs that Aaron had set up these original method files that we're still using, but they're 12 Thomson windows. There's the range there, and we have 50 windows. And again, injecting 100 nanograms of either JJN3 or HeLa peptides in there. I'd use non-staggered windows for both experiment because Mike doesn't use staggered windows. So I compared those. And then I wanted to compare the significant protein IDs between these two instruments. And so this was a little bit disconcerting. So the Venn diagram here shows the difference that the overlap here is about 16%. So there's about 1,500 significant IDs from the Exploris and about 780 from the QE, but those two lists overlap only 320 of them. I think so.
How are you defining significant difference?
Just ran it through ProteoDA and the significance is based upon the fold change and the p-value, whatever that is. In what is it?
Yeah, it's twofold change and then a 0.05 p-value.
Yeah, 0.05,
Are the,
right.
I'm not sure if you're using adjusted or not, but anyway.
Mm-mm. Yeah.
I think this is really driven by the fact that the explorers identified a lot more proteins. I think that the overlap is small because it, you know, identified 800 more proteins.
But those are the different, it's not the total.
That's right. These are not total. These are the ones. So because that's
Right, but it identified a lot more, and therefore it identified a lot more that were changing because it identified them.
okay.
Yeah, the windows were different. The acquisition windows were significantly different, right?
Mhm.
Not as different as it looked. That was the MS2 mass window
Oh, was it?
range. Yeah, this is 400 to 1,000 on the MS,
But you all use 12.
but it was.
Oh,
12
did I
Thompson
get
versus-
down the wrong
Yeah,
window
but it was-
there?
it
Oh,
was
okay.
12 versus 20, and that's just due to the speed of the instruments.
Yeah. So I did as all scientists do. I verified this by doing this again. So here's the volcano plot difference between the Exploris and the QE there. And there is one interesting. Is this the one that has the interesting data point sticking way out there? Well, anyway, I did this again, only this time I did it between the Exploris and the Eclipse. There, the numbers of proteins identified are much similar. When we do our DIA, we can do DIA on the Exploris, or we can do it on the Eclipse, and we get comparable results. They're not exactly the same, but they're much more similar. And the overlap here is a little bit better. And so here you've got the Eclipse comes in with a little bit more significant IDs at 1,600, and the Exploris is 1,530, but the overlap there is about 700 and what's that say? 791. Yeah. Yeah. I don't have answers. I just did the experiment, and it may be that the way I searched the data or did the experiment or something.
Well, this is also why I try to tell people that want to compare experiments months later,
Yeah,
try to run everything together and not at separate experiments.
yeah, right.
And I think this tells you get the same platform.
All those technical differences.
Mm-hmm. Right.
I think a lot of fold changes are around two. They're like, you have more low fold changes than you have large fold changes. Right, and so they're
Like, oh,
putting noise
close, yeah.
in a quantitative ratio. So a lot of, I think, your difference in your Venn diagrams are things that are fluttering around two fold change.
Right.
2.1 in one sample, 1.9 in the other sample. It's now looking like all or nothing, whereas if you did a scatter plot. Between the two, you probably actually have fairly good correlation, but they'd be, wouldn't be tight right on that axis.
Yeah, and
Okay.
I think the others
That's
are
a great idea.
probably teetering on being detected or not detected. And I think that the differences are either great or they're near the line. I agree. And so I think significance, yeah, and you can see how populated the data is on the small fold change, just kind of meeting significance there. So yeah, that's a good point.
So what you?
Yeah, I was gonna say the same. I mean, same thing. So we did the experiment similar to what you did, different samples, but between the timsTOF and the HT and the 480, and when you set sort of standard things like you did here, we saw the same thing. Maybe like 20% overlap, and then you're like, man, this. So which instrument do I use? What I trust? And you're like, and then, but then you we did the exact same thing. So then we set a minimum signal for detection and raise the fold from 2 to 2.5. And then we get like 88% overlap. The numbers go down, of course, but then you start to believe, and then it comes down to, and then we start using that for our real reporting. You know, what is that? Because really what we see is that if for DIA, when you start getting down into the lower signals, I mean, I don't know how much to trust that, but I'm sure with that same data set, you could just manipulate some analysis factors and probably get that closer to like a 70-75% overlap just by employing some of those methods to bring that up.
That's great. Thank you guys for weighing in on that. Again, as you guys can imagine, with the symposium coming up, I thought, oh, I'll have plenty of time to do all these experiments. I'll have plenty of time to get all this data analyzed. This will be great. And then as it got closer and closer, I said, or we can all just look at the data together and try to make sense of it. Okay, so all right, I've got one more little vignette here, and that's about database searches. Okay, so I wrote this months ago, and so these are kind of cheeky things, but I said, DIANN, it's free, it's functional, but there's no visualization. Spectronaut, it's pricey and easy. And then this PD, I said what most people use for DDA, and I said Chimeras is needed for DIA. And then scaffold DIA, we use that exclusively for all our DIA stuff until it crashed hard, like in July. And we had multiple communications back with them, and they were just like, and because our data sets just get bigger and bigger and bigger, and it really struggles when you have a lot of samples. And we started using Spectronaut, and it did. I mean, we're getting crazy. We're opening up 4 or 5 searches all at the same time on the same computer. That thing is just going nuts. So trying to do the same thing, I want to compare QE data, 100 nanograms, this same experiment here, searched by Spectronaut and DIANN. So, are we getting, just like you're saying, it's platform specific. Is it database, software specific as well? Obviously, you saw earlier that the numbers are not the same between all of these different players here. And so the DIANN searched data is over there on the right, and then the Spectronaut searched is over there on the left. And I tried to keep, the settings as similar as I could. I did use the direct DIA searching for Spectronaut. And this, so comparing the significant protein IDs between DIANN and Spectronaut for this particular thing, and we get a similar sort of thing. We get about 783 for one, and we got 529. And as you might, the more, I believe DIANN is bigger. I didn't put that on there. Yeah. And that goes along with what everybody's been saying, is that DIANN gets you more stuff. And then the total proteins there, no surprise, we ended up with about 10% more with DIANN than searching Spectronaut.
Yeah, and so I want to chime in on this. So this is 100 nanograms on a QE. And, the chromatography is not crazy. I mean, these are what? The chromatography is normal chromatography. So it's not, it's not like, a 50-meter long column with 1 micron particles. I mean, this is something people can do, right? I mean, it's, and the numbers are a little higher here than they would be with just a cell lysate, because you're picking off the abundant proteins from the 2 different cell lines. Mm-hmm. But it's still, I don't know how many it would be if it were just one of the cell lines, but probably 3,000, 3,500 or something like that. So this shocked me. I was shocked. I was expecting maybe this from the 1 microgram, but this was surprising to me.
Dennis, I have one question. Maybe she will answer tomorrow. if you wanted to customize databases in DIANN, it's easy to do it because suppose that you're working with species that are not very well known or very popular. It's something that in DIANN, how you can upload it, you can prepare for different place, how you can run DIANN with, for example, certain bacteria or certain fungus or whatever. It's a way to install a database or customize a database and put inside.
Yeah, any database you can
Fast
download.
save file, right?
But you need to get it from ready the database from the source, or you need to take and work on it to create a spectral library or something else.
No,
No.
just a FASTA database you download
Okay.
from Uniprot or something, yeah.
And there aren't even any. What surprised me is there are no experimental parameters. There's nothing about collision energy or it's just it just takes those sequences and generates its spectral library.
I wanted to add something to make more happy to Mike. I make an experiment in DDA comparing QE plus versus Floris 240. And the QE was better. Sorry guys for the term, but really the 240. Something similar, you know, that was better, even though it's all instrument. Similar condition, everything.
Okay, so I'm gonna move on. I've got one more here. And this one goes back to, I alluded to just a second ago, we switched over from encyclopedia or using scaffold DIA and to Spectronaut. And so we had some question as to, we used to do gas phase fractions, but now we don't, because with whatever edition 17 or whatever of Spectronaut, the direct DIA has just made it to where we don't feel at this time that instrument time is well spent making the gas phase fractions. So, but I wanted to go back and I wanted to compare that. I want to do an experiment where I compare the number of protein IDs for a project searched by encyclopedia or scaffold DIA and Spectronaut. And so this is just tells you, gas phase fractions were collected. We did 6 of them, 100 Thompsons at a time from 4 to 500, 5 to 600, et cetera. We would pool all of the samples and then we run those first. And there's the information about the conditions that were set. So the encyclopedia workflow looks something like this. You take your gas phase fractions collected, and then you create this chromatogram library, and then you search your sample files against those. And the output from scaffold DIA for this particular, this is a different study. It was actual tissue. It's carotid artery from a mouse model. And it was one that we had back in July. And we got about 3376 proteins here with a 1% FDR. And if we did Spectronaut, where I made a library from the same gas phase fractions. So all the files are exactly the same, just using a different database searching platform. You can see that we ended up, I was actually surprised because for some reason in my mind, I just knowing this group that I came into that we're a little we're a little stingy here. We don't want to have elevated numbers. So I thought we would be using the one software program that would give the smallest number because we were going to be the most confident about that. But actually, in this case, using those gas phase fractions, Spectronaut didn't do as well. I mean, it only got 2,949 proteins there. The information over here on the right is the stats about the spectral library. So of course it has to find all this information about the spectral library, and then it takes your samples and then searches against that. And so somebody had asked that question earlier, why do you need that? Why not just search it against the whole FAFSA database? And it has to do with that false discovery rate, getting to that 1%. If you search against a really big library that contains everything in the world, then you're going to find stuff that isn't really there. And so being able to limit that library down to something that is only what's you expect to find really helps. So then the last one that I did here is or the next one is I did you can also in Spectronaut, you can make a library from the sample files. So this is kind of an intriguing thing. And I still haven't quite got my head wrapped around it, but instead of doing, the gas phase fractions, the reason why that makes sense is because you are, you're smaller windows, you're much more focused. And so you would get more detailed information there. But the nice thing about making a library from the sample files is you don't have any more instrument time, right? You only run the sample files once, and then you use those same things to make your library, and then you turn around and search against them, which sort of seems like cheating to me. It's like double dipping or something, but the protein groups are about the same, 3,100 there by doing that. And then the last one here was doing the direct DIA approach, which Spectronaut is so famously good at. And now our protein groups jump up by 25%. And so, and this is kind of what has been sort of preached here for years as we've come along with DIA, and that is, yeah, getting more IDs is not necessarily better. Especially when you go interrogating the data and you're really looking at what's there. But then the other side of that is, well, we're doing a global proteomic experiment, right? And the whole idea is to throw everything up against the wall and see what sticks, right? We're looking for leads, trails. So, but that's the, so I use the word conclusion here, but I just said that, we're using, based on this experiment here, we use a Spectronaut or Encyclopedia in creating the spectral library from the gas phase fractions or the samples themselves. Basically, you got the same number of protein IDs, plus or minus 10%. It was only when you went to that direct DIA that we suddenly saw this big jump, and I didn't validate any of those findings beyond that. So, but I just thought that was kind of an interesting experiment for me to be able to go through so that I could, try to understand how these subtle changes in the workflow change things, downstream and all that. Comments?
Thanks, Dennis, for going through that. That's a lot of work, and you've really that kind of convinced me about the merit of staggered workflows in this day and age. Like, is it worth going through all the extra trouble when the instruments nowadays are capable of acquiring data at that speed? But with this growing number of protein IDs with experiments, like whose responsibility is it for the accuracy or the trueness of the data? Is it something that core directors take on and sign off on, or is it something that encourage the downstream collaborators to follow up on?
I think this is where Andy's comment about this being the Wild West, and it depends on who the local sheriff is, right?
I was afraid of that.
Yeah, absolutely.
I have a comment.
Especially in DIANN, DIANN's kind of a black box to me, right? There's very few parameters you can change. You often don't know how it's working. So if you use staggered windows, I assume that it would actually use the overlap to its advantage, right? Like it's using machine learning to optimize the results in very fancy ways. But at the end of the day, you're kind of just left trusting this black box, and then you get a spreadsheet output, right? No visualization of the data, as you've mentioned. Whereas Spectronaut, I would say has probably an order of magnitude more dials to its detriment to some extent, right? It takes a while to become an expert with Spectronaut. And even if you read, I think, the 100+ page manual, we still have questions on a regular basis that we have to fire off to them. A lot of the terminology they use isn't necessarily standard or I'm familiar with. And so kudos to you for being able to even compare these two and get the parameters as close as possible. But what can you say about the additional features Spectronaut brings? One of the things I'm often struggling with is it provides imputation, right? And one of the things you're looking for in DIA experiments is completeness of data. And so it's not perfect though in my hands, like you still have missingness.
It can, I mean, you can turn it on or off. I mean, it can, right?
Yeah,
But you don't
but-
have to use
And
it.
there's 3 different
Yeah.
ways it does imputation.
That's right.
And the part of the reason, and the only reason I use it sometimes is it will do imputation that's based on a sample basis or even based on the noise around the individual peak that should be there or could be there. Which
Yeah.
probably, it's like,
That's probably the best that you could hope for, right? Because
yeah,
you can't detect it. And if you could detect it, it would be at that noise level. And so
Right.
it gives you a multiplier so that at least, Lima would handle them all the same. And that was my question earlier, because sometimes you don't know what you're leaving off the table. So.
yeah, there's many times where you see signal, but you don't see an ion envelope and you don't see signal noise of 3. So you say, I don't have it. But using that little bit of noise is better than just a raw imputation with randomness. So I would, that's one of the advantages I think in Spectronaut, but I haven't done the side-by-side comparison with all the permutations. It would just take you forever.
Yeah, personally, I turn off the imputation. I don't want it in my data.
Yeah.
Because then I'm dealing with that on the back end when I do my analysis. Then
Yeah,
I can do whatever I want with the numbers. But my problem with Spectronaut right now is trying to go from the peptide level back to the protein and how they're actually matching up their numbers. I can't backtrack it.
yeah,
So you have to, because you do have to go peptide by sample by each individual protein. And I even pull those out and try to do it the way they say they're rolling up and it doesn't match.
yeah.
Can't
We've
get
had
it
that
over.
same experience. Yeah,
And so they don't know, well, I know the support person, they don't know the machine learning algorithm at all. They don't know how it's working.
Yeah.
But it's also proprietary and I do machine learning, so it's really bugging me that they won't even give you a realm of which machine learning tool we're using.
right.
So I'm trying to track that down and figure out how that's working, because
Yeah.
it's a problem. Because I also don't know, they have the different options and the default is set to the mean for the roll up for both
Yep.
of those, which if you got an outlier, that's
Yeah.
not good.
Yeah.
So, and then I found out they've got, so if you set automatic for the IDs, that's the max LFQ method for max quant.
Yeah.
And the other one is the quant 2.0. They said that's the top 3 method, but it can't be just top 3 because it's their own version
Mhm.
of top 3. So there's a lot behind the scenes that can't even backtrack.
Yeah. And it
Also a black box.
Yeah,
Yeah.
Diane's, similar, right? So
Yeah.
it's doing top 3, but they don't say how they do it when you don't have 3.
Yeah.
Or they may say it, but if they say it, it's buried somewhere that I haven't found or can't understand. That's probably more likely.
You're supposed to be able to set the number in Spectronaut, so it doesn't have to be 3, it can be whatever you
Yeah.
set.
I think maybe just one thing to mention too is because machine learning is being used and these are models, if you run the same data set a second time, you don't get the exact same result.
Right.
And so that,
Interesting.
It becomes kind of my standard. If there's a protein that I run every algorithm at it and it's still different, then that's an important one. So if it drops off just because I changed the normalization method, then I'm probably not going to spend a lot of time in the lab looking at it.
Yeah.
Right.
It's just kind of, that's where I draw the line for the biologist who are trying to look at it.
Yeah, Dennis and I were talking yesterday about these, the plots of, DIANN or Spectronaut on significance. And I said, well, what we probably should do is have 2 outputs, run it on both and report the union of the significance and the intersection and, start with the intersection. If you
Yeah.
can't find an interesting biological story there, then back up to the union, right? Or look at the union at your hypothesis that you were going to go after, but then the intersection is likely where the real differences lie. Statistical, significant aside, there's, I've always said this, you got statistical significance and you got real.
Yeah.
Real ones are going to be in that.
The software that I want to show tomorrow, it's a software that I just bought for the data science core for Embry. But they actually run multiple algorithms and they have a table and it stars it if it passed all of the algorithms or not, which is very, very helpful. So